Good morning to one and all. Today we're going to talk about virus, its morphology, structure and reproduction. Coming to the definition of virus, virus is a Latin word meaning poison or venom. It was generally believed that the virus or poison were carried in the night air and caused by many unexplained disease. It is an infectious microbe consisting of a segment of nucleic acid. It either has DNA or RNA, only one, either DNA or RNA, surrounded by a protein coat. Viruses that attack bacteria were first observed in 1915 by a British scientist Watt. Later in 1917, French investigator de Hallert fully studied that these bacterial viruses and named them as bacteriophages or phages. Now, viruses can be defined as small obligate intracellular living form containing only one type of nucleic acid. Either it can be a DNA or RNA. Earlier, they were also called as ultra microscopic viruses because they could not be seen with a light microscope. Later, they are called as filterable viruses because they can pass through a very fine filters which holds the bacteria back. Now they are simply called as virus and a mature virus particle is called as virion. Now coming to the morphology of virus, there are different shapes and different sizes of viruses. You can see this elaborated picture. There are different sizes of viruses. Example, you can say hepavirus, rabies virus, measles, HIV, SARS, papillovirus, Ebola virus, dengue virus, hepatitis, etc. All viruses are extremely small and cannot be seen with a compound microscope. They measured in millimicrons and vary considerably in sizes. They ranges from 15 millimicrons to 450 millimicrons, and among that the smallest is foot and mouth diseases of cattle and the largest one is parrot fever. In humans, the smallest virus is of yellow fever and the largest is pox virus. Coming to the shapes, they are different. It can be many shapes and sizes, but can generally classified into four groups: either helical shape, like it is a form, long form, filamentous structureless forms. Polyhedral includes icosahedral and prolate shapes. Envelope shape have an envelope. Head and tail, which means have an elongated isohedrons and helical tails. The shape of the virus is determined by how its structural protein, depending upon the protein shelf, assemble to form a protective protein coat called as capsid. The capsid can be single or double and it is made up of few or many types of structural proteins. So you can see examples of here. A rabies virus is of bullet shaped, TMV is rod shaped, polio virus is spherical in shape, pox virus is rectangular or brick shaped. Some of them are irregular in shape and bacteriophages have head and tail like sperms. Coming to its different examples, you can see they are all spherical. They are coronavirus, adenovirus, reovirus, retrovirus, influenza virus, parovirus, and epidenoviruses. These are some of the examples. Now, let us know about the important structure of viruses. It contains three parts: the nucleic acid, capsid, which is a protein coat, and sometimes an envelope. All the viruses doesn't have envelope, only few of them. They do not possess any cellular organisms and hence they are called acellular. So there are no cells like other microorganisms. This means they lack most of the cell organelles like plasma membranes, ribosomes, etc. Hence, they may usage of the host machinery in order to replicate. They only contain one type of nucleic acid, as we said, either DNA or RNA. Let's talk about the nucleic acid. It contains only one, DNA or RNA. The central part of the virus contain a nucleic acid. If you see the picture, the central part, that is a viral genome, either it can be one type, RNA or DNA types. It is mostly getting the viral information about the viral protein. The DNA containing virus like examples chickenpox, hepatitis B and herpes virus, and RNA is like hepatitis C, measles and rabies. Coming to the capsid, the main genome is surrounded by a protein coat called the capsid. In the picture you can see the middle uh, shape, it is a capsid. It provides a definite shape to the viruses. The subunits of protein called as capsomers combines to form capsid. With all capsomers, they are forming a capsid. Each virus has a specific number of capsomers in the capsid. For example, herpes virus 162 capsomers, while adenovirus has 252 capsomers. Now, what is envelope? Some animal viruses have another covering outside the nucleic acid and and capsid, that is nucleic capsid, which is derived from the host cell membrane called as the envelope. If you see the picture, the round part is an envelope. Virus having no envelope are called as naked virus or no envelope. Now, envelope protein. On the surface of the envelope, if you see a round shape, the outer layer round shape, it has a viral glycoproteins which are present called as envelope protein. These projections are called as envelope protein. They help in identification and binding to the receptor site of the host cell membrane. And they are also helpful in protecting virus from the host immune system. Now coming to the multiplication and replication of virus. Viruses multiply but much more complex process than binary fission. It cannot reproduce in inanimate media, but they can only grow by inoculation into animals or embryonic eggs or tissue cells. So they cannot live outside, they require a host for their reproduction. They lack the machinery to synthesize their own proteins. Instead, they synthesize the proteins from the machinery of the host cells. The phenomena of viral multiplication was first studied in the bacteria and later in animal cells. Although the pattern of multiplication in bacteria and animal virus is much similar, but there are also important differences. So let's see. As we say, cultivation, it is a method, is a type of research often used to distinguish viruses that can cause disease. The specimens of a contaminated person's bodily fluid are accumulated and mingled with cells that would be used to generate a virus. If no infection spreads to the cells, the virus culture is said to be negative. If a virus that can induce a condition dominates a cell, the virus culture is called as positive. So let us see the various methods. One is animal inoculation method, the second one is cultivation in chick embryo, and the third one is the cultivation in tissue culture. For example, in the animal, the mouse is the most commonly used animal inoculation method for virus isolation. In this method, it is one of the oldest methods for the growth of viruses. In this method, animals are inoculated with viruses. The virus grow in these animals in the form of a disease. The second one is embryonic egg or cultivation in chick embryo. First, Burnett method was later used to cultivate virus in numerous places of embryonic egg. So in this method, chick embryo at a suitable stage of development, that is 5 to 10 days, it is inoculated with the virus. A small hole is made in the shell and the virus material is inserted with a needle or a capillary pipette in the hole. The hole is then closed with the paraffin wax and then the egg is re-incubated. Now the virus multiplies in the cells. The third method is cultivation and tissue culture. Cell culture is the most commonly used during diagnostic virology for virus cultivation and the assay was performed. In this method, many tissues of animals, including human, can be grown outside the body in the nutrient fluids. When the tissue growth has reached the desired stage, it is then inoculated with virus. Now the virus starts growing in the tissue. Now coming to the replication of viruses, there are 7 stages. Usually, unlike bacteria, many of them which can be grown on an artificial nutrient media, but virus requires a living host for replication. The 7 steps or 5 steps you can say they are attachment, penetration, uncoating, replication, assembly, maturation, and release. Stages. So let us see individually. The first stage is attachment. If you see the picture, how the virus is trying to enter the host cell, that is called as attachment. The virus becomes attached to the cell by a specific cellular receptor, which can be glycoprotein, phospholipids, or glycolipids. In second stage, it is penetration, or also called as entry. The virus can enter the cell most commonly via receptor mediated endocytosis method. This is same process by which many hormones enter the cell. If you see how a virus is entering into a host cell. The third one is uncoating and replication. Once inside the host cell, the viral capsid must be uncoated to release the viral nucleic acid. Uncoating may be achieved by host or viral enzyme that will degrade the capsid. Once uncoated, virus, that is either DNA or RNA, replicate by switching the host machinery from cellular protein synthesis to viral synthesis, and the viral proteins are produced. In third, fourth stage, assembly. New synthesized viral protein are post-transcriptionally modified and packaged into a virions that can be released from the infected host cell to infect other cells. And finally, it is a release. Virions are released from the cell either by lysis or budding. In lysis, the infected cell dies and the virions are released. In budding, the virions take some of the host cell's membrane with it as it leaves. This is normal, does not kill the infected cells. This is how a replication takes place. Hope you understood, girls. Thank you.